Welcome, everyone. I'm delighted that you can all join us for our fifth and final presentation in the Osher Lifelong Learning Institute 2025 at the U Lecture Series. I'm Laura Peterson, an OLLI volunteer. The Osher Lifelong Learning Institute at the University of Minnesota is a vibrant learning community for people aged 50 plus. Part of the College of Continuing and Professional Studies, OLLI belongs to a national network of Osher Lifelong Learning Institutes supported by the Bernard Osher Foundation. I would like to acknowledge and say thank you to our sponsor, the University of Minnesota Alumni Association. We acknowledge that the University of Minnesota Twin Cities is built within the traditional homelands of the Dakota people. It is important to acknowledge the peoples on whose land we live, learn, and work as we seek to improve and strengthen our relations with our tribal nations. Today's lecture is Cutting Edge Cancer Therapies. Our presenter is Dr. Christopher Pennell. Dr. Pennell received his PhD in genetics from the University of North Carolina at Chapel Hill. He is a professor in the Department of Laboratory Medicine and Pathology at the University of Minnesota Medical School. Dr. Pennell is an immunologist, a member of the Center for Immunology, and the Masonic Cancer Center's Associate Director of Education. The floor is yours, Dr. Pennell.
Thank you, Laura. Thanks again for the invitation to speak to you all. And I'm going to focus on cutting-edge cancer therapies, but I'm going to start with a story that you might be familiar with. This is Emily Whitehead, and this picture was taken on her 5th birthday in May of 2010. 2 weeks after this picture was taken, Emily was diagnosed with a type of white blood cell cancer called pre-B-cell acute lymphocytic leukemia. Her parents were devastated by the diagnosis, but were heartened by the prognosis because her chances of long-term survival were about 85 or 90%. So Emily went through the standard chemotherapeutic regimen, but unfortunately, even though she was put into remission for a short period of time, her disease recurred, and it got so bad that she was considered terminal and was only given a couple of months to live. At that point, her parents enrolled her as the first child in a clinical trial at the University of Pennsylvania. And the picture on the right is Emily this year. She's a sophomore at UPenn, and she has been cancer-free for about 13 years. This was all due to a type of cancer immunotherapy that I'm going to talk about towards the end of my talk. But this is the power of cancer immunotherapy. It has saved lives of patients that would have died otherwise. So the Masonic Cancer Center is one of only 57 comprehensive cancer centers in the country. And we're considered comprehensive because we have research and education and clinical activities that span the entire cancer continuum. To put therapy into perspective, though, I need to spend a few minutes talking about etiology, which is how cancer occurs. Because with this knowledge, then you'll have a better understanding as to how the therapies I will discuss actually work. So cancer arises from the corruption or the misuse of genetic information. And genetic information is DNA, deoxyribonucleic acid. It's a mouthful, I'll just call it DNA. And this is a biochemical information and storage and retrieval system. The entire collection of DNA is called its genome. OME, that suffix, is used to denote the entirety of something in biology. So genes are chunks of DNA that encode for information, and all of that is called the genome. I like to think in pictures and use analogies. And so analogies for the genome include your computer's hard drive or an owner's manual or a cookbook, some source of information that contains pretty much everything you need for a particular project. And using this analogy, then, one file or one recipe is equivalent to one gene, which is a unit of information used to produce a discrete product. And in the case of biology, one gene encodes a protein. So the protein is the molecule that does the work that's encoded by a gene. And the word gene was coined, I think it was like 1909 by a Dutch botanist. And gene derives from a German word for origin. And so this this cartoon depicts 3 different cells in our body, muscle, liver, and let's say a white blood cell. And for one cell to divide, it has to copy all of the recipes in its family recipe notebook. So each of these cells, even though they perform different functions, have the same set of information. And as I said, all of that information collectively is called the genome. This book contains 23 pairs of chapters or chromosomes. A gene, as I mentioned, is a unit of heredity, easy for me to say, or a particular recipe. Because all cells need to perform similar functions, generate energy, make membranes, they all use the same set of genes, and we call these housekeeping genes. But what distinguishes a muscle cell from a liver cell from a brain cell is that those cells use different recipes in that recipe book, and these are called tissue-specific genes. The entire book contains almost 13 billion letters. There are only 4 letters in the DNA alphabet, A, C, G, and T, but each book contains 12.8 billion letters. And for one cell to become 2 cells, every letter in that book has to be copied. And so I'm going to paint a picture of enormous copying potential, and not potential, but enormous copying that occurs in our bodies every day. So it's estimated that the average human adult makes about 300 billion new cells every day. These would be white blood cells, cells in our intestinal tract, skin cells, cells that need to replace dying and dead cells. So if you multiply 300 billion cells per day times 12.8 billion letters in the DNA alphabet per day, you come up with a number that's 3.84 trillion billion letters are copied every day in our body. If you were to stretch this amount of DNA out into a single line, it'd be over 400 miles long, which is roughly 4.5 times the distance from the Earth to the sun. So when you're dealing with numbers this astronomically large, mistakes are going to happen, and this is how cancer arises. This is a cartoon of how a cell functions and how it accesses that information in its nucleus, which is the organelle that houses the recipe book or the DNA. So a cell gets a signal from its environment. These satellite dishes represent receptors, which are ways the cells can communicate with the outside world, if you will. So a signal is delivered, it's transmitted.
Yeah, baby.
To the nucleus. Now the nucleus knows what recipe to access, but the recipe book contains precious information. It's protected by this membrane. And so the information stays within the nucleus, but it's copied. And then that copy is transmitted to the factory that translates the genetic information into a molecule, a protein that can do the work.
I'll get you, my pretty, and your little dog, too.
So it goes from the DNA to RNA, which is ribonucleic acid, to proteins. And this is the central dogma of biology, this flow of genetic information from DNA to this messenger RNA that takes it to the protein. DNA and RNA are basically the same language, the language of nucleic acids, and the letters in this language are called nucleotides. Proteins, though, speak a different language, and their letters are amino acids. So the transition from DNA to RNA is called transcription, but the change from RNA to protein is called translation, because now you're going from one language to another. And hopefully all of this will make sense in a few minutes when I put it together with respect to therapy. So how can a mutation in information lead to a change in the cell's behavior? Well, as I already mentioned, genes encode proteins, so it follows that altered genes would encode altered proteins that now are not functioning properly. And if we look here on the top line, this is the language of DNA. These are nucleotides. There are only 4 different ones, but the order connotes the language of the protein. So if you have 3 A's in a row in DNA, it translates to an amino acid called lysine. 3 G's in a row translates to an amino acid called glycine, threonine, alanine, and so on. And so in this case, my diagram shows that this amino acid lysine at the left end is positively charged. Aspartic acid is negatively charged. So you can imagine that in the entirety of this protein, these two amino acids would attract one another, and the intervening amino acids would loop out. And this loop might serve some sort of function, like perhaps a docking site for another antibody that can come on and recognize this loop. Or maybe this loop is involved in some enzymatic function. But it's the structure of the protein that dictates its function. And the structure is dictated by these amino acids that comprise the protein. So now let's say a single mutation occurs where this first A is now changed to G. Well, what happens is now GAA, like GAC, encodes aspartic acid. So we've changed from a lysine, which was positively charged, to an aspartic acid, which is negatively charged. And now these two amino acids would repel each other and destroy that loop and alter the function of the protein. So that's how a single mutation or change in a language can result in a change in behavior and function of a protein. And it's these changes that drive cancer. And what I'm going to I'll tell you the punchline right now, the immune system can detect these changes. And what we're going to talk about is how the immune system is used to treat cancer. But to put this all in context, the way that cancer arises is through, as I said, the change in DNA, mutations, or misuse of DNA. And these mutations allow cells that we have evolved to prevent cancer from occurring to overcome those barriers. That sentence was not, I didn't phrase that correctly, but mutations allow cells to overcome barriers that we have evolved to normally prevent cancer. So in this diagram, this cell down here is going to divide into two cells, and each of those has the identical recipe book as the parental cell. Another cell divides, but now this solid cell represents a mutation that occurred during that copying process, and that allowed it to overcome this first barrier. And then over time, this process is repeated, as I said, 300 billion cells a day. And over time and years, eventually enough mutations can accumulate such that cancer can become clinically apparent. And this is why cancer is more prevalent in older individuals than younger individuals, because this accumulation of mutations occurs over time. So that's the background I wanted to give with respect to how cancer arises. And I particularly wanted to make the point of mutations changing the amino acid sequence of a protein. So now let's talk about therapy. Actually, I should step back and just briefly define what each of these components in the continuum are. So etiology is how cancer arises. If you know what causes cancer, if you can avoid it or prevent it, you're doing yourself a favor. An ounce of prevention is worth a pound of cure. If we know what we're looking for, we can catch cancer early, maybe after it's hopped over one or two of those barriers where it's more easily treated than it would be after it jumped over the last barrier. And that's what happens when a tumor becomes clinically apparent and we have to treat it with therapy. Survivorship, many patients are now surviving cancer, but they often have health problems related to the chemotherapeutic cocktail that killed the cancer cells. And then palliative care is for patients who are terminally ill. So I'm going to focus on therapy right now. So cancer therapy has evolved over the millennia. Prior to the 1900s, surgery was our only option. And many patients died from infections after surgery than from the disease itself. But we know that surgery has cured many patients of cancer. At the turn of the last century, radiotherapy and traditional chemotherapy came into play. And at the turn of this past century, we have now what's called precision therapy, which I'm not going to discuss, and immunotherapy. And what's driven this evolution is specificity. So specificity is the holy grail of cancer therapy. If you can kill a malignant cell and not any healthy cell, even the healthy cells from which the tumor were derived, you can give a therapeutic dose of the drug or the treatment to cure the disease without killing the patient. And so specificity is what's driven this evolution. And now I'm going to focus on immunotherapy because this has revolutionized the field of cancer treatment. So as the name implies, immunotherapy is using the immune system to treat cancer. And it's useful because it has 4 characteristics. And these include specificity. I mentioned a couple of minutes ago that the immune system is exquisitely specific. So if you have 2 proteins that are 1,000 amino acids long each, and one of them is mutated such that there's only 1 amino acid difference between those 2 proteins, the immune system can detect that difference as being foreign and eliminate the cell that made it. It's incredibly potent. So 1 killer white blood cell can kill thousands of tumor cells. It's long-lived, it has a memory. So if the tumor ever comes back, the immune system is poised to eradicate it before it becomes problematic. And it's adaptable. One of the characteristics of cancer is genetic instability. So even though almost all cancers are derived from a single cell that went rogue, when that cell divides and divides and divides, it is genetically unstable, meaning that there are subtle differences in all of those progeny with respect to one another and the original tumor. The immune system can move, it can basically move along with the tumor, if you will, and it can adapt and it can eradicate those newly forming tumor cells. So it's these 4 characteristics that make the immune system really attractive to treat cancer. And immunologists have known about these characteristics for over a century, but it's only been within the last 15 or 20 years that we've been able to take advantage of these characteristics to treat cancer. And I'll tell you why we weren't able to do it until recently. Before I do that, though, I need to give you an Immunology 101 quick talk to set the stage. So it will make, the therapies will make sense if you understand how the immune system functions in a basic sense. So the primary function of the immune system is to maintain balance or equilibrium or homeostasis in the body. So here we have, and when the immune system becomes activated, when the body experiences injury, infection, or disease. And the end result of this activation and subsequent deactivation is a return to a healthy state. And this occurs in a physiologically normal and healthy individual. So the wound is healed, the infection is resolved, the disease is eradicated. Well, one of these functions of the immune system is what we call immune surveillance. And this refers to the immune system's ability and characteristic ability to remove newly forming tumor cells before we even know we have them. And this probably happens on almost a daily basis in our body. And so if you think about that series of brick walls that I showed you a few moments ago, I think of the immune system as the last brick wall, the last barrier that is eradicating malignant cells before they become problematic. And so this is my highly technical cartoon showing this. So the immune system is constantly eliminating nascent, newly formed tumors. And as I said from a moment ago, from animal studies and from anecdotal studies of immunosuppressed humans, we know that this happens on a regular basis. So how does the immune system become activated and how does it recognize foreign molecules? So this is my Immunology 101 talk briefly. So if you become infected with a virus or a bacteria, those pathogens, disease-causing organisms, release molecules into our body that we cannot make ourselves. We don't have the enzymology to make them. We've evolved ways to recognize those molecules as not being self and being indicative of an infection. Collectively, we refer to those as stranger signals. The counterpart to that would be internally derived danger signals that come from the contents of cells that normally don't see the light of day. So if a cell is injured via a burn or a cut, it will release its intracellular contents. And normally those stay inside the cell and they're not outside the cell. But if they get outside the cell, they again are recognized as being a cause of some sort of danger. Something has happened that the immune system needs to take care of. And the immune system is multi-layered, but from a 30,000 foot point of view, you can divide it into 2 arms. First responders and second responders. The first responders are like EMTs or firefighters. They're on the scene quickly, and they can basically take care of many types of infections or problems. But as they're taking care of that, they're delivering a call to arms. And that's calling in second responders. And these are specialists that can take care of a specific infection or a specific type of disease. And together, those will eradicate the source of the strange or the danger signals, returning the individual to equilibrium. So the two classes of immune cells, white blood cells, that form the first responders and the specialists are called innate and adaptive cells. And innate cells, this is just a cartoon of a particular subset of innate cells called eosinophils. And if you look at eosinophils in a particular tissue, in the skin, for example, they all express the same set of receptors. So they're at a barrier organ, the skin or in your gut, that is likely the site of the first infection. And they're there on site, and they've got receptors that can recognize stranger or danger signals. And so once those signals are delivered, these cells can respond. The specialists are called B cells and T cells. These B cells come from the bone marrow, T cells come from the thymus. They're colored differently because they each represent a different molecule. So each of these has a highly specific receptor that will only respond to a particular molecule from, let's say, E. coli versus Pseudomonas. So this may respond to E. coli, this may respond to Pseudomonas, this may respond to COVID, and so on. So they're highly specialized, but they're very effective, much more so than many of these innate cells. So what do these cells recognize? The term we give them is called antigen. And this is derived, it's a hybrid word derived from anything that can generate an antibody, so an antibody generator. So back in the 30s and 40s and 50s, immunologists were immunizing every animal they could find with different molecules to generate antibodies. And collectively, they gave what they jabbed into these animals the term antigen because it was an antibody generator. We didn't know about T cells back then. But when we identified T cells and knew that they were highly specific, we used the same word antigen. But antigen is an umbrella term that is used to describe thousands, if not tens of hundreds of thousands of different molecules. The commonality is that they can be recognized by either a B cell or a T cell. So what do B cells do? And again, this is all related to the types of therapies I'm going to talk about in the last 30 minutes. So B cells function as an extracellular surveillance mechanism. So if you get infected with a bacteria, for example, most of those don't go into your cells. And so it's outside of your cell, extracellular. And the way that B cells take care of this is by secreting proteins that will bind specifically to the pathogen. And these are called antibodies. And so these different colors represent different B cells, and they all express antibodies on their cell surface. And if they become activated, they can spit out these antibodies and secrete them. So they're in the serum and in your blood and in the fluid in your tissues and the lymphatics. And these antibodies have the capability to bind specific pathogens, disease-causing entities, and eliminate them. And so these are different viruses and bacteria. The antibodies can bind to them, but you see that only certain antibodies bind certain pathogens. And when they bind them, they're basically marking these cells for destruction by the immune system. And so the virus is eliminated, the bacteria is destroyed, and again, you return to homeostasis. This is a key consideration that you need to understand to make the immune therapies that I'm going to talk about to make sense to you. So when you immunize an animal with a complex protein, there are multiple sites on it, multiple bumps. So if this is a big protein, you're going to have a lot of different structures on the cell surface. And the B cell, we can make antibodies against many of these surfaces. And this is called a polyclonal response. So many clones of B cells are making these antibodies. And they're really good to take care of the antigen. But therapeutically, you can't use this because it's not reproducible. If I immunize myself with a protein, chicken ovalbumin is a standard one. I immunize myself with that. I'm going to make certain antibodies, Laura's going to make different antibodies. Grayson is going to make different antibodies. And if I immunize myself again 5 months from now, the antibodies that I make 5 months from now are going to be different than the ones I made today. And so the lack of reproducibility really limited our ability to use these therapeutically. But then we had a technical revolution in the mid-70s, which allowed us to make what are called monoclonal antibodies. So let's say this antibody is the one you really want, because let's make believe that this molecule that I'm calling an antigen is a virus. And this part of the virus is what binds to the cell and gets it into the cell. So if you can block that binding site, you can neutralize the virus and not allow it to infect the cell. And so that's the antibody you want. And so how do you get it? Well, we can take that B cell that makes that antibody and clone it. And so we can generate billions of these cells that are all genetically identical to one another and are all making the antibody that we're interested in. And this is called mono for one, clonal, because it's clonally derived from a single progeny. And so these monoclonal antibodies are essentially homogeneous proteins and can be treated like a chemical. They're reproducible, so I can make these antibodies in my lab. Somebody can make these antibodies in Japan, and they're identical to one another. And so this is what revolutionized the field of immunotherapy. And then it evolved. So initially, back in the 70s, we can only make these monoclonal antibodies from mouse B cells. And originally, anti-cancer monoclonal antibodies were made in mice and injected into humans. The problem was that we recognized this mouse protein as being foreign, not from us. And we made antibodies against this mouse antibody that eliminated it. And so this problem was resolved through a series of steps. So this region of the antibody at the tips of the arms are what specifically bind to a bacteria or a virus. And this is what we're interested in, this specificity. So what we could do genetically is to take these regions of the mouse protein and graft them onto human proteins. And these are called chimeric antibodies. So the blue represents the human, the maroon represents a mouse. A further refinement came when we realized that only parts of these mouse proteins actually contacted the antigen. So this, if you look at it three-dimensionally, looks like a barrel with loops coming out of the top of the barrel. And it was those loops coming out of the top of the barrel that actually contacted the virus or the bacteria. So what we could do is we could just take those loops that had the specificity we wanted and graft them onto even more human genes. And this is called a humanized antibody. But now for the last 15 years or so, we've been able to make fully human antibodies. And these are the names, these are the suffixes that you'll see. If you're looking at commercials on TV, you'll see something that says UMAB. MAB stands for monoclonal antibody. U means it's fully human. If it's XI, it's a chimeric antibody. So the next family gathering, you can, show that you're one of the immunology cognoscenti by telling them, oh, that's a human antibody if you see it on TV. And it's big business. So these are the number of antibodies that have been FDA or EU approved over the years. And you can see that in red. These are cancer therapies. And so cancer has really played a dominant role in the development of these monoclonal antibodies, and it's a billion-dollar industry. I'm going to come back to how antibodies have been used therapeutically in a moment. Now let's look at the other type of first responder, a T cell. And again, this is called T because it develops in the thymus. Instead of surveilling what's going on outside of the cells, it surveys what's going on inside the cell. And you can see how this would be important if you consider how a virus works, many viruses. Viruses work, they're basically molecular parasites. They get into a cell and they take over this DNA, they take over this protein making machinery. So now the cell's protein making factory is making viral proteins, and you have tens of thousands of viral particles inside a cell, and then the cell will rupture or release these viruses. So what a T cell does is it can recognize a virally infected cell and kill the cell before that virus producing factory has a chance to release tens of thousands of viral particles. And it does this by recognizing bits of proteins that are called peptides. A protein is called a polypeptide. So a peptide is maybe 8 amino acids, 10 amino acids of that protein. And these are displayed on the surface of the cell. So proteins are made by the cell. They're broken up. Bits of the proteins are displayed on a platform called an HLA molecule. And this is what a T cell sees. It sees bits of proteins displayed on a platform on the cell surface. And if these proteins come from if these peptides come from our normal proteins, the immune system recognizes them as being self, and the cell gets a check mark and it's good to go. If this protein is either a mutated protein in a cancer cell or a viral protein or something that is not us, the T cell can recognize this and kill that cell before the cell has a chance to wreak havoc. And this is just a cartoon showing this. So the T cell receptor recognizes this peptide bound by this platform molecule, and then it can kill the tumor cell. And this is a video showing how that happens. Let me get out of here. Oops.
These are immune cells called cytotoxic T cells. Cytotoxic T cells are able to recognize and kill virally infected and cancerous cells in the body. These T cells are on patrol, searching for their targets. Let's take a closer look at how they move. Cytotoxic T cells are constantly migrating around your body. They move through your tissues by pushing out the membrane at the leading edge of the cell and pulling themselves forward. When a T cell encounters a cancer cell, an explosion of membrane protrusions explore the surface of the target. Cytotoxic T cells kill their targets with toxic chemicals. The chemicals are housed in these red structures called lytic granules. You can see the lytic granules polarized from the rear of the T cell to the blue cancer cell. These toxic granules secrete specifically toward the target, protecting innocent bystanders which may be nearby. As the membrane of the target is compromised by the toxins in the granules, our indicator flashes bright red, signaling the impending death of the cancerous cell. Movies like these allow us to study the efficiency with which cytotoxic T cells operate. Now we'll leave you with a few more movies of T cells doing what they do best, killing cancer.
And that music really happens inside your body. That's the best thing about it. So if you hear, this sort of swelling of music, your immune system is functioning. So as I mentioned before, one of these surveillance mechanisms of the immune system is to eliminate newly formed tumor cells. So with that definition then, a clinically apparent tumor has escaped recognition. And I've just depicted that by this barrier, this brick wall. And it was the composition of this brick wall that stymied the field for about a century. We didn't know how tumors evaded the immune system. But due to basic research within the last 20 years, we have a much better understanding of the bricks in the mortar that are used by the tumor cells to erect this barrier. And with that information, we can overcome this suppression. And that's what I'm going to talk about now. And this is the genesis for how the first strategy for overcoming immune suppression mediated by tumors was achieved. So back in 1990, early 2000, most immunologists were trying to figure out how to activate an immune system to generate better vaccines and so on. So this diagram here shows time and days on the x-axis and the number of disease-causing entities. Let's say this is a bacteria in a dotted line, and then the number of T and B cells that can respond to it. So as you get more and more, as the infection goes on, the immune system can respond, and ultimately it reaches a point where it can eradicate the disease. and then it has to turn itself off. And for years, we were focusing on the left side of this graph. How do you turn on an immune response? But Jim Allison and a few other people were interested in how do you turn off an immune response? Because if you don't, you can have sequelae such as autoimmunity. And so turning it off is as important as turning it on. And what Jim Allison found was a particular molecule that was used to turn off the immune system once the stranger and danger signals were gone. And then the leap of logic that it gave him, that it enabled him to receive the Nobel Prize in 2018, was that he thought, I wonder if tumor cells have figured out how to put the brakes on the immune system, just like it happens in a viral infection. And the answer was yes. And so you can think of this as a gas pedal and a brake pedal. So the gas pedal activates the immune system, the brake pedal shuts it down. And Jim Allison found that this brake pedal was used by tumors. And so collectively, these molecules that break or suppress the immune system are called checkpoint molecules. The one that Jim Allison found is a molecule called CTLA-4. He then made a monoclonal antibody that prevented CTLA-4 from suppressing a T cell. So this is a tumor cell on the right, a T cell on the left in green. He made an antibody that interrupted this breaking process. And then he treated mice with this. And the mice that did not get the antibody died from the tumor. The mice that did get the antibody survived. And now there are a whole bunch of these checkpoint molecules that have been identified, and antibodies have been made to either them or the things they bind on T cells. And this has just exploded the field. And so the way that I think about this is that we've basically been able to reduce the integrity of the wall. And now the immune system can activate, can find and kill the tumor cells before they become problematic. And an example of somebody who's benefited from this was Jimmy Carter. When he was 85, he was diagnosed with stage 4 malignant melanoma, which is typically a terminal disease of, it's a terminal skin cancer. He was treated with this checkpoint blockade therapy, one of these monoclonal antibodies, and it allowed him to survive another 15 years. You can't read all of this. That's not the point. The point is to just overwhelm you with information showing that there are many, many of these checkpoint inhibitors that are used in many different types of disease. And these have been game changers because, like for President Carter, they have allowed patients who were terminally ill, otherwise terminally ill, to live. All right, I'm going to leave antibodies for a little bit and talk about some types of killer cells. And there's another type of killer cell in addition to a cytotoxic T cell that was in the movie called a natural killer cell. And work here at the Cancer Center by Jeff Miller and his colleagues is exploiting the ability of natural killer cells to kill tumors such as prostate cancer cells. And so what Jeff and his colleagues have made are molecular bridges that make it much more likely for the natural killer cell to bind and find the cancer cell. So these molecules are called trikes. KE stands for killer engager, so it engages a killing cell. Tri means it's got 3 components. One arm binds something specific on the tumor. One arm binds something specific on the killer cell. And the third thing provides fuel to the killer cell to allow it to function better. And by adding these, you can really enhance the ability of these cells, these killer cells, to find and kill tumor cells. And so that's the outcome. This is a movie showing a blue killer cell that's going to kill these live cancer cells. So what they did was they added cancer cells and the NK cell to this well, plus that trike. Without the trike, you wouldn't get killing of the cancer cells. With the trike, you do. The second point of this is that this natural killer cell is a serial killer. So you see one killer cell has just killed 3 tumor cells, and it can continue to go on killing tumor cells until it can no longer find any. And so by injecting these drugs into patients, you can take advantage of their own immune system and by basically allowing the immune system to find the tumor cell much more readily and to respond better. This is not limited to just prostate cancer. So these are head and neck cancers. The red circle in the middle represents tumor cells that have been stained with a red dye. The area around it are proteins that the tumor cell has made to make its environment conducive for its growth. NT means that this is not treated, so these are just the tumor cells themselves. These have been treated with just one of the three components, the gas of that trike, and this has been treated with the trike itself. And there are natural killer cells in all of these, okay? So if we look at here, over time, the tumor cells pretty much are nonplussed, right? They're growing, there's not a problem. If we throw in the growth factor for those killer cells, these clumps represent proliferating populations of killer cells, but they're not having much of an effect on the tumor. But if we put in the gas plus those engagers, now that you can see these dark areas, these are the natural killer cells that are finding the tumor and are eradicating it. And so these sorts of studies in the lab have led Jeff and his colleague to do a number of clinical trials. And these are the different types of cancers that they're trying to target here at the U, using a trike that has one arm that binds the killer, the gas is the second, and then the third would be specific for each of these different types of cancers. And those specificities are derived from monoclonal antibodies. So the arm that binds the tumor target is a chunk of the monoclonal antibody that was developed. The second way, so I've presented now to you two ways that we can use the immune system to treat cancer. One is using antibodies by themselves. The second is using components of antibodies to form a bridge between a killer cell and a tumor cell. The third way is that we can genetically modify the killer cells to kill what we want them to kill. So here, these are cytotoxic T cells. The different colors represent different specificities. These were the things they were born to see. We can genetically reprogram them to see what we want them to see, which in this case is a tumor-associated molecule. And the way that this works is that we take somebody's white blood cells out of them, genetically engineer them, expand them up to billions in the lab, put them back into the patient. And then this is like a tsunami that's breaking over a seawall. So there are so many effector cells that there aren't enough suppressor molecules to keep them in check, and they can kill the tumor. And this was the strategy that allowed Emily Whitehead to live today. So how do we genetically modify them? We make what is called a chimeric antigen receptor. Chimeric is the adjectival form of the Greek of the word chimera, which is a mythical beast in Greek mythology, the head of the lion, the body of a goat, the tail of a serpent. And so this is a molecule that we put into a T cell. Outside, so this is the outside here. This is a part of an antibody, and this has been chosen because this part of the antibody will bind to something specifically expressed on the tumor. And then we take that and we genetically link it to molecules that can activate the T cell. And so this is part from a B cell. This rest is from a T cell, which is called a chimeric antigen receptor. We genetically put that into T cells. And now this allows the chimeric antigen receptor to work. So the way the process works in the clinic is that we have a cancer patient. We can take their white blood cells. We can genetically modify them, and we put them back into the patient. I'm not going to talk about the bottom part. That's another strategy that's used. But this is it. So we take out blood cells, genetically engineer them, expand them to big numbers, put them back into the patient. And this type of therapy, again, has been a game changer. As I told you, it saved Emily Whitehead's life. So the person who led that clinical trial is a colleague named Carl June. He's at the University of Pennsylvania. He said that if Emily had died, the field would have ground to a halt. And the reason is that for pediatric clinical trials, if the first child enrolled dies, nobody wants their child to be the second child in that trial for obvious reasons. So even though this doesn't work for everyone, it works for 60% of patients that are treated. All of these patients were terminal, though, remember, but it was fortunate for Emily and for the field that she was one of the survivors. So the summary is that we can get 80 to 90% durable, complete responses in patients with a type of malignancy and these patients would have died otherwise. But with every therapy, there are side effects. And I'm going to talk just briefly about one of the side effects, which is neurotoxicity. And the reason I'm going to talk about that is that you're a captive audience, and this is what I do. But I've only got 1 or 2 slides, so don't worry about that. And this type of toxicity was totally unanticipated. And the reason that it's important is that we can't release a patient from the hospital until they're cognitively back to normal. And so even though the neurotoxicity can range from something as mild as disorientation to something that's fatal, like swelling of the brain, even if a patient is mildly disoriented, they can't be dismissed from the hospital because they may walk in front of a bus or cause some harm. So my colleagues and I developed a mouse model to try to understand how this works. And what we do is we take the skulls off mice, glue on windows, and then we use this bar to secure the mouse to a block. And then we shine a laser into the brain to see what's going on with the cells. And we take advantage of genetically modified mice. So we have green mice, red mice, blue mice. So we take T cells from a green mouse, genetically modify them, put them into a red mouse, and it makes it easy to see. So this is a movie from the brain of one of these mice. These green cells are the genetically modified CAR T cells. This is a blood vessel. You can see the red blood cells going through. There's a green cell that just zipped through there. And so the green cells are getting out of the blood vessels in the brain, and they're parked here. And so what I'm doing with my colleagues is we're trying to answer several questions. Like, how did they get there? Who are they talking to? What are they saying? And how are they causing problems? So this is one of the cool things that I'm doing. So yeah, in the last minute or two, I just wanted to just summarize what hopefully I've said. And that is that immune surveillance constantly eliminates nascent tumors. And so by definition, cancers have escaped immune surveillance. The characteristics of the immune system that make it attractive for cancer therapy include specificity, potency, memory, and their adaptability. The therapeutics that are derived from the immune system that we use to treat cancer include these monoclonal antibodies, immune cell engagers like the trike, these adoptively transferred cells that we genetically modify like CAR T cells, and vaccines, which I didn't have time to talk about. And the success of these cell-based therapies, in fact, all therapies, is critically dependent on overcoming cancer-mediated immune suppression. And so it was this barrier that prevented us from exploiting specificity, potency, memory, and adaptability for years. But now that we know how the wall is built, we don't know all of the components, but we know many of them. We can now either destroy the integrity of the wall or genetically engineer enough cells to swamp over it. And all of this type of research is being done here at the U, which is it's a fantastic place to work. So hopefully I didn't put you to sleep or bore you, and I'm going to stop sharing now and see if I can answer any questions if they exist.
Oh, that was fantastic. Thank you. So the very first question right out of the chute, you can probably anticipate, which is, will your work be affected by NIH cuts from the Trump administration?
Yes. So I'm going to only work on white male mice from now on instead of black mice. But I'm a co-investigator on a grant that is written to attract underrepresented students in the biomedical research field to cancer. We've had it's a longstanding program that I've had for about 15 years. We got a perfect score on the review, which has never happened to me. But the person at the NIH who was shepherding this was just fired. And so we don't know if we're going to get the money or not to continue the program. So for my outreach, yes, it's definitely affected. For my own research, it's not. But many of my colleagues' research, it is affected. People that are looking at cancer health disparities, for example, or education efforts. Yep. So yes, the answer is yes.
Yeah, okay. So I should have mentioned just before I started asking questions, I wanted to mention quickly to the audience that Dr. Pennell is not able to answer your specific clinical questions about your specific disease states or people that you know. He is a researcher. He works with lab mice. He is not a clinician. He doesn't work with humans. So I just want to put that out there. When you hear that I'm not asking your specific question, that's why. Okay, lots of questions about this wonderful therapy of CAR T cells, but we all know that these don't work in everybody. Why?
There are a few reasons. One, it depends upon the person's health. So we take white blood cells out of an individual. If the person has gone through, 30 rounds of chemotherapy and stuff, the starting cells just may not be healthy enough or viable enough for us to expand in the lab. So that's one reason. Another reason is that the tumors may lose the target antigen that the CAR cells see. That's called antigen loss. So if they turn off the molecule that the CAR T cells are looking for, then they're basically like stealth tumors, if you will. So antigen loss, the person, him or herself, and then sometimes in the lab, for reasons that are still not clear to us, we can't grow up enough cells to give back to the person. And then finally, the cells themselves sometimes become exhausted, and that's an actual term that we use because they no longer can kill after 1 or 2 rounds of killing. And so, as you can imagine, these are all areas that are under active investigation to try to overcome.
Yeah, cancer cells are extremely devious, it seems to me. And even though we finally discovered how to target these receptors on the outsides of them, the fact that they can actually withdraw those receptors so that they're not visible anymore is amazing.
It is. It's like it's almost like a predator-prey relationship, and it's evolution in a microcosm. So if you have, let's say, 1 billion tumor cells, and 10 of those billion have undergone a mutation that turns off the target molecule, and then you treat it with something that's going to target that molecule, you'll kill, like 99.99999% of the tumor cells. They all go away. The person is in remission, but that small subset was not touched because it didn't express the target. And then over time, they can expand. And now when they reach a certain level, they become clinically apparent and problematic, but they no longer have the molecule that was targeted to begin with. And it's this genetic instability that is the issue. And so there are some strategies that we're using rather than trying to target one of these molecules at a time. We're trying to target 3 or 4 or 5 of them at the time, with the thought that it's very unlikely for any one of these tumor cells to lose all 5 at the same time.
Yeah. Okay. Do these therapies that you've mentioned today typically only work on early stages of cancers?
No, they don't. So they work on late stages of cancer as well. So Emily was stage 4. Jimmy Carter was stage 4. And in fact, they're only used on patients who have failed every other therapy. Actually, the CAR T cell therapy is used for patients that have failed every other therapy. But there's a move now to try to treat patients at earlier stages. So yeah.
Yeah.
But the checkpoint inhibitors, those monoclonal antibodies that block the break, they can be used not in stage 4, but earlier stages of cancer to try to prevent them from progressing to more deadly later stages.
Okay. Do you know, is an immune therapy considered the first line of treatment for cancers now, or is chemotherapy and radiation still used first? And when you fail that, you move on to these.
It's a mixed bag. So some therapies, some immune therapies are used as a first line therapy. They are few and far between, though. Mostly it's because the oncologists have much more experience with chemotherapy and radiation therapy because they've been around for decades, whereas immunotherapy is relatively new. As I mentioned, neurotoxicity. So there are these toxicities that we don't anticipate that pop up oftentimes in the immune system. And so most immunotherapies are not used as frontline therapies, but some of them are. And
Okay.
I can't tell you, which I can't give you specific examples of which ones are first line therapies, but well, having said that, I guess I can. For some autoimmune diseases, there are soluble proteins that mediate an autoimmune response. And now we have antibodies that can block that. And so those can be given, right up front for some autoimmune diseases like arthritis or eczema or things like that.
Yes, thank you for addressing that. There were a number of questions about whether or not this could translate to autoimmune therapies. So
Yep.
perfect. Is cytokine storm a risk for these therapies? And if
Yep.
so, how is that addressed?
Yeah, so let me define that and then I'll speak about it. So cytokine storm represents or refers to basically systemic inflammation. So when you put billions of these killer cells into a patient who has billions of cancer cells, what happens is when the killer cell binds to the cancer cell, not only does it kill the cancer cell, but it releases those call to arm molecules that I mentioned, and they cause inflammation. And one of the sequelae of inflammation is permeability of the blood vessels. And so if the blood vessel becomes more permeable, white blood cells can get out of it and into the tissue to fight the infection. So a local inflammation is good. So if I cut myself here, it will get red, it'll swell. So that local inflammation is really good because it's bringing in all of the white blood cells to take care of the infection that occurred at the site of the cut. If it occurs systemically, it can kill you because your lungs can get leaky and you can literally drown. You can get swelling of the brain where the water presses in on the brain and will destroy the brain. So that systemic inflammation is called a cytokine storm. Cytokine is the name given to these molecules, these call to arm molecules. And so with CAR T cells, Emily almost died from systemic inflammation. But Carl and his colleagues realized that there was one of these molecules that was playing a key role, and they were able to use an immunotherapy to suppress it. And this actual immunotherapy was used for treating autoimmune diseases. And so they were clever enough to realize that if they could suppress it, neutralize it, that the inflammation would go away. So yeah, so cytokine storm and neurotoxicity are the two major toxicities associated with CAR T cell therapy.
And can you reduce the risk of cytokine storm damage by surgically removing most of the tumor burden before these therapies?
Yeah, that's a good idea. Most of the CAR T cell therapies, though, are for what we call liquid tumors, which are leukemias. And one of the reasons they're used for leukemias is because the white blood cells are malignant, and we're also using white blood cells to kill them. So they're in the same environment. So you can't surgically remove a liquid tumor that's in the bloodstream, right? But for solid tumors, yeah. So there would be probably a debulking and a variety of other things that would happen. And we don't know if the cytokine storm is actually going to happen with solid tumors. We just don't have enough experience with that just yet. My bet would be that it wouldn't be, and it's going to be more problematic with leukemias that are systemically distributed throughout the body.
I see. But is CAR T therapy still of some benefit to patients with solid tumors?
Those are in clinical trials right now. So I think the answer is yes, but it's been a mixed bag. So there are currently 7 FDA-approved CAR T cell therapies. They're all for hematologic malignancies, which are malignancies of white blood cells. We don't have any FDA-approved CAR T cell therapies for solid tumors. Having said that, what's FDA-approved is about 10 years behind what's being tried in the clinic and in the lab. And there's a 10-year gap where it has to go through all of these clinical trials and safety things before it becomes useful or before it becomes widely available. And so what the FDA approves may seem cutting edge, but for people like me, it's 10 years out of date. And so for solid tumors, we've had some success in mouse models, some limited success in people. And the major problem seems to be identifying what are you going to target. So ideally, you'd want to target a mutated protein that's only expressed on a tumor and not on the healthy cell from which it was derived. Those are hard to find, and there are only a couple of examples of those that exist. And so you have to be careful, because when you're killing B cells for these CAR T cells that like that saved Emily. She had a B cell leukemia. It killed all of the leukemic B cells, but it's also killed all of her healthy cells, her healthy B cells. So she is devoid of antibody producing cells, so she's immunocompromised, and she has to receive antibodies pulled from other people for the rest of her life. You can't do that strategy with a liver, for example, because if you target healthy liver cells, you're dead. So that's the conundrum, and that's what people are trying to figure out right now.
Is there a move to replace or supplant bone marrow transplantation in these patients with hematologic malignancies with CAR T therapies instead?
Yeah. Yes. So this is I'm in a gray area right now.
Yeah, sorry.
No, that's okay. So let me just explain really briefly the rationale for a bone marrow transplantation. I mentioned at the outset that the holy grail of cancer therapy is specificity. Chemotherapy kills rapidly dividing cells, but it does not distinguish between healthy cells that are rapidly dividing and malignant cells that are rapidly dividing. And so what happens is if you give a high dose of chemotherapy, high enough to kill all of the tumor cells, you're going to wipe out rapidly dividing cells, which include cells in your bone marrow, which are the cells that give rise to all of the cells in your blood. So the way that this rationale for bone marrow transplantation is that you give a really high dose of chemotherapy, hoping that you kill all of the tumor cells, knowing that you're going to wipe out the bone marrow. And after a couple of days, when the chemotherapy is flushed from the body, then you replace the dead and dying bone marrow cells with some from a healthy donor. Okay. And included in that are the stem cells or the precursor cells that give rise to all of the red cells and all of the white cells. So can CAR T cell therapy be used in place of that? What happens now is that somebody would have to have failed. For the clinical trials, they had to have failed bone marrow transplantation. Right now, honestly, I don't know if it's used in lieu of bone marrow transplantation, but I bet it is. Sorry, that's the best I can tell you.
No, that's great. I did sort of go across that line of clinical versus laboratory medicine.
No, that's fine. I mean, I should know, but I
That's
don't.
okay. If one checkpoint inhibitor medication fails to kill the cancer cells, can a different medication with two checkpoint inhibitors be used?
Yep, exactly. And so those are the things we're looking at. So yeah, so you try one, and then if that one doesn't work, you can switch to another, or maybe you can try two or three. But each one of those uses has to be approved by the FDA, because if you put two or three together, individually they may have certain toxicities, but synergistically they may have a totally new toxicity that you would not be aware of unless you did some sort of clinical trial. So yeah, so there are the two favorite ones are called anti-PD-1 and anti-PD-L1. Those are used pretty regularly for many malignancies because those are two key ones. But if both of those fail, there are a couple of others that are being investigated. Yeah.
So you can definitely combine various strategies,
Yeah.
immunotherapy strategies, I should say.
Yes. And so what we have to do, what scientists try to do is minimize the number of variables so you can better understand the results, right?
Yeah.
But cancer doesn't work that way. Cancer will, as we mentioned a few minutes ago, will mutate and will lose these target molecules. So there's this combination therapy that is now being tried. But again, you've got to try each one individually to understand what the pros and cons of each one are separately. And then you've got to test each combination together to find out what the pros and cons are. And then if you've got three together, it just starts going like this with difficulty. But that's the way the field is going.
Okay.
Combination therapy.
Okay. One listener says some cancers have a lower mutation burden and may not respond well to immunotherapy. Is there a linkage between mutation burden and response?
Yep, that's a very astute question. So there are some tumors that we call immunologically cold and some tumors that we call immunologically hot. And that is directly related to the infiltration of white blood cells in the tumor itself, and that's related to the amount of mutations. So if we look at melanoma, melanoma is a skin cancer. Melanoma is caused primarily by ultraviolet light. Ultraviolet light causes many, many mutations. And so if you look at the number of mutations in melanoma tumors compared to pancreatic, the number of mutations is 2 orders of magnitude higher. And melanoma is hot tumor. So there are a lot of targets for the immune system to identify and kill if the melanoma hadn't erected that barrier. And so immunotherapy, as I mentioned for Jimmy Carter, can, those checkpoint blockade therapies can remove that barrier. And now you've got killer cells that can recognize 20 different mutated molecules and kill the tumor really well. Pancreatic cancer is more of a cold tumor. And so there aren't a lot of targets that we've identified. And it also creates a much more immunosuppressive environment. So not many T cells can get in. And when they get in, they're really suppressed heavily, and there aren't a lot of targets for them. And so again, these are active areas of research trying to figure out, can we make a cold tumor immunologically hot by getting the cells in there? And can we identify targets for them once they get in there?
Interesting. Okay, someone is asking for the FDA treatments that are approved, are they standard now, or does someone, like, you can get them pretty much anywhere in the US, or do you need to go to a special research clinic to gain access to them?
You have to go to a special research clinic to gain access, and the reason are these toxicities that I mentioned. And they're not ready for, outpatient administration right now, because the cytokine storm, for example, that has to be treated aggressively, quickly, and you have to know what you're doing, or the patient will be dead within 12 to 24 hours. So because of those toxicities, they're limited right now to tertiary care hospitals like our Cancer Center, Mayo Clinic, and other places like that. The immune checkpoint blockade can be done on an outpatient basis, because there are fewer toxicities associated with that, and they're not acute. They don't occur very rapidly.
Yeah.
And these are expensive. These CAR T cell therapies, $300,000 to $500,000.
I was just going to ask you a question about that.
Yeah, they're not cheap.
Do you know how patients pay for them?
Hopefully through insurance. But no, I don't know how they pay for them.
Wow.
Yeah, and because making the CAR T cells is expensive. You've got to harvest the cells, ship them out to some place. They do the genetic engineering, and all of this has to be done under what are called good manufacturing procedures, which means everything is clean, everything is quantified, and it's all careful because it's going back into a person. So there's an expense there. Then they've got to be shipped back to the hospital where the patient is. This takes a couple of weeks. And then the patient is infused, and then many of them develop cytokine release, so that has to be squashed down. If they develop neurotoxicity, they may be in the hospital for another, several weeks. So all of that combined leads up to that high price tag.
Yeah. Are you able to say whether or not the risk of death from the treatment is higher with these immunologic treatments than it is with chemotherapy? Do you have any knowledge on that?
For the CAR T cell therapies, these are terminally ill patients, so everything we've tried has failed. So they would have died otherwise. In terms of the other types of therapies, the immunotherapies, there's checkpoint blockade and antibody-based therapies, I think they're much more tolerable than chemotherapy, because they're much more specific. As I mentioned before, chemotherapy kills rapidly dividing cells. So this may be of interest, or may not, sorry, but the length of DNA in an individual cell, if you were to stretch it out into a single molecule, is about 7 feet or 2 meters long. It's packaged into an organelle called the nucleus. That's 1/5000th of an inch in diameter. So you've got 7 feet and 1/5000th of an inch diameter structure. That's a heck of a packaging job. For one cell to become two cells, it has to unwind that DNA so the enzymes that will copy it can access it. So rapidly dividing cells have more of their DNA unwound or exposed, if you will, than a cell that is not dividing. And so when you deliver the chemotherapeutic drug, it's cleared by your body within a couple of hours. But during the couple of hours of exposure, cells that have more of their DNA naked are going to be modified by that chemotherapy. The DNA is going to be broken up and the cell is going to die. So that's why rapidly dividing cells are hit by chemotherapy, but it doesn't distinguish between a malignant and a healthy cell that are rapidly dividing. Whereas the immunotherapy takes care of the specific, it's exquisitely specific, and it only acts on the cell that has the target for which the antibody is specific. So I would think that most antibody-based therapies are much more tolerable than chemotherapy.
Yeah, sounds like it. Can you speak to the progress made with allogeneic CAR T-cell transplants as opposed to autologous?
Not knowledgeably.
Okay.
So autologous means they're coming from your own self. Allogeneic would be from somebody else. And anytime you put somebody else's cells into your body, there's going to be what's called a graft, host versus graft response. I'm going to recognize Laura's cells as being not mine, and I'm going to try to eliminate them. These patients are immunosuppressed, so the allograft may work well. But I think the underlying premise to that question is, can you make an off-the-shelf reagent, or can you make something that's more readily available than and so you wouldn't have to make it specific for each person. You could take something off the shelf that was already qualified, and stamp of approval was given to it. And the answer to that is yes. Jeff Miller and his colleagues are working on that with the natural killer cell approach. And Bo Weber and Brandon Moriarty and David Lagerspada here are working with that with T cells as well. So the goal would be to have something that's off the shelf that you could give rapidly. And I think we're not there yet. We probably won't be there for another decade or so, but there's definitely progress being made.
Yeah. All right. Can you address CRISPR and gene editing?
Yeah, so the way that we currently make these CAR T cells, there has to be a way that you get the genetic information, that the genes, the DNA that encodes the CAR protein into the killer cell. 15, 20 years ago, when this field was first emerging, the state of the art was to use viruses, because viruses are very effective genetic delivery vehicles. So we use viruses to get them in there. And the viruses are attenuated, so they don't proliferate themselves. They just serve as a delivery vehicle for the CAR DNA. The problem with this approach is that the viruses can integrate that DNA quasi-randomly in the genome. So if you disrupt a key gene, you may be causing problems that you weren't aware of, or you couldn't control. CRISPR is like surgical precision. You can identify exactly where on the chromosome you want to put that CAR gene. And so scientists have identified what are called safe harbors, which are regions of the DNA that you can modify without any known adverse effect. And so what CRISPR-Cas9 allows us to do is to insert the CAR gene into these safe harbors where they won't disrupt any normal cell function. And again, that's on the horizon. So in 10 years, the CAR T cells will be made with CRISPR to introduce it. But right now it's using this viral approach.
All right, thank you. What about the potential someday for vaccines for cancer treatment or cancer prevention?
Yeah, so that's a reality now. So prophylactic vaccines prevent disease, therapeutic vaccines treat disease. We know several viruses are responsible for certain malignancies. So human papillomaviruses, 16 and 18, I think, are the subtypes, are responsible for about 90% of cervical cancer. So if you get vaccinated against HPV, you reduce the risk of becoming infected, and therefore you reduce the risk of developing cervical cancer, and some male-associated cancers as well. So HPV is a prophylactic vaccine. Epstein-Barr virus, there's HPV, excuse me. HPV causes certain types of liver cancers. So again, if you get vaccinated against the hepatitis B virus, you decrease your risk of becoming infected and developing cancer. EBV is another virus. So those are prophylactic viruses. Therapeutic viruses are just starting to become effective now. And the reason that there has been a delay between the efficacy of prophylactic and therapeutic is that therapeutic is trying to raise an immune response in an immunocompromised individual who's been compromised by the disease that they have and perhaps prior treatments. And so, if you think about that wall again, rather than starting on the ground floor with a therapeutic vaccine, you're starting from a hole. So you've got an even bigger burden to overcome. And again, the problem is, what are you going to target? How are you going to deliver it? But and I was doing work on DNA-based vaccines myself for cancer, and we had some results that were positive, but they weren't strong enough for me to consider it a viable alternative. But other people have been working on it for decades, and there are now platforms similar to the COVID vaccine, mRNA vaccines, that so this is really cool. Sorry, there was a paper that just came out a couple of months ago where what they did was they isolated RNA from the tumors from 30 different people. And then they identified the bits of the proteins that that mRNA would encode that could be presented on those platforms to T cells. So they put all of those RNAs in a package and delivered it to patients. And this is a 5-year study now. And they're finding that if they deliver these specific vaccines to specific people, they can get some positive responses. So hopefully there will be therapeutic vaccines that arise. I mean, this is a bit of a divergence, but bear with me. So Angelina Jolie is probably known to most of you. She's an actress. She had inherited a mutated DNA repair gene that meant that when she was in her 50s or 60s, she had close to a 90 to 95% chance of getting breast or ovarian cancer. She decided to have a prophylactic double mastectomy and hysterectomy after she finished bearing children. And she did that because of those numbers. I mean, that's a horrific decision to have to make. And if we had a vaccine that could prevent that from happening, that would be wonderful. And that's what people are hoping for.
So the prospect of a vaccine is not limited to those cancers that are triggered by a virus.
No, because what you want to do is you want to identify what's called a neo meaning new, epitope. Sorry, the field is replete with jargon. You want to identify a mutated protein that is in a cancer cell. And hopefully that mutated protein contains an amino acid that can be recognized by the immune system when it's put on the cell surface by a T cell. And so that's what that paper that I alluded to a few moments ago did. They used an algorithm that predicted what these mutated proteins would do in an immune environment and then tested them, and they're finding that they're getting some positive results. So yeah, so viruses provide non-self proteins, viral proteins. Mutations provide non-self proteins, which are our own selves, but they're mutated.
Okay, I'm sorry. I just have to ask one more question about that. Go just a tiny little bit deeper. So there are 20 proteins in the human body. Is that right? Or
20 amino acids, you mean?
20 amino acids. Okay, so yes, so the proteins that might be expressed on a cell that you might be targeting with a vaccine could be very, very unique to the type of cancer that.
Yes.
Okay, got it.
Yeah, so you can think of a protein as like if you think of a train, a railroad car, right? You've got the connections between the cars, and those are called peptide bonds. And each car would be an amino acid. And if you take the train off the track, the positively charged cars would attract the negatively charged cars. The cars that were hydrophobic would attract hydrophobic. So there are all of these physical characteristics that allow a protein to fold into a particular three-dimensional conformation, and that dictates its function. And so if you mutate one of those, you may or you may not alter the function of the protein depending upon where that change is. And so for a viral protein, pretty much all of those cars in that train are different from ours. I mean, the order is different, how they're arranged is different. And so the immune system is going to recognize that as being different. But for us, a single mutation, the train is pretty similar to a healthy train, right? If there's only one or two cars that are different, it's a it's a harder target to find.
Okay, that makes sense. Thank you. Someone here is asking or saying that has been some news in the last few days about immunotherapy as a treatment for some solid tumors. Are you aware of that?
Yeah, so some of these, some of the checkpoint blockades, that big list that I showed you that you couldn't read, many of those are solid tumors. So immunotherapy using monoclonal antibodies has been effective for a number of solid tumors. Melanoma is a solid tumor. Cell-based therapies are the ones that seem to have a better effect right now on liquid tumors, just because the modified cells can find the tumor cells more easily in the vasculature than they can. One of the reasons that pancreatic cancer is so difficult to treat is that it's almost a perfect storm of disease characteristics. It makes a wall of protein around it. So it's almost like a capsule that really impedes cells getting in there, drugs getting in there. In addition to that, the cells in that environment there's crosstalk between healthy cells and the pancreatic cancer cells. And the pancreatic cancer cells tell the healthy cells to modify their behavior to make the environment more conducive for the tumor cell. And so now the tumor cell can get growth factors and things from the healthy cells, and then the healthy cells also limit the amount of white blood cells that can get in there. So it's a nightmare of a problem, which is why it's so difficult to treat. That was a bit of a tangent, but anyway,
No,
immunotherapy for antibodies is certainly used on solid tumors, and it will be used for cell-based therapy more frequently as we overcome these barriers.
yeah, I know pancreatic cancer is also a concern because it doesn't produce symptoms, I think, until it's fairly
Yeah.
advanced. Yeah, and so just
Yeah,
a
we don't
triple.
we can't. So in that cancer continuum, prevention and screening, we, we don't know really what a lot of the causes for pancreatic cancer, although we have a few clues. And then the screening part, we don't have any markers that say that there's a problem brewing, not like PSA for prostate cancer. And, you can do mammographies and radiographic screening for breast cancer. So we don't have an early marker for pancreatic cancer. So we only know about it when it's really, really far down the progression pathway.
All right, now someone is asking about Yamanaka factors. Is that something that you want to talk about, or
Now, I've heard about them, but I don't know much about them, to be honest with you.
yeah, don't either. All right, do you know if Keytruda is an immune system
It
medication?
sure is. It's a monoclonal antibody. If you
Ah,
look, Keytruda is the brand name, but if you look in parentheses, it'll say blah, 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 UMAB at the end.
mm.
So, yep.
Okay, and then I think I have one more question to ask you, and so I just want to remind the audience to put in your questions if you have anything quick, although I can always hope that Dr. Pennell will be back with us again next year to give us more insights and maybe more success stories about what's happened in the preceding 12 months. The question, and you may have answered this a tiny little bit already, or maybe quite a bit, but what future therapies are in the works?
Using CRISPR-Cas9 to genetically modify cells, not for cancer, but well, it is going to be used for cancer, but it's already been used to cure sickle cell anemia in some patients because there's a genetic mutation that gives rise to sickle cell anemia. And so you can take a patient's bone marrow out, modify those cells, get rid of the bad bone marrow, if you will, do a bone marrow transplant with the gene corrected cells and eradicate sickle cell anemia. So that's been realized. In terms of what's on the horizon for cancer therapy, these cell-based therapies are really garnering a lot of interest because as we know more about the cell's capability and as we learn more about how we can modify it, we can really take advantage of the cell's natural ability to kill tumors, but really tweak it to turn it on in certain cases and turn it off when we don't want it on anymore. So those are big things. Monoclonal antibodies are huge, as I've mentioned before. There's another area of, it's called patient-specific medicine, which is called precision medicine, and that's based on pharmacology and genetics. So we would sequence your DNA from your tumor, sequence your healthy DNA, identify based on the tumor mutations what drug to give you, and then if we have a choice of 5 or 6 drugs, we would then use the information about your own healthy enzymology to figure out what drug you're going to tolerate best, and then give you drug 3 out of a choice of 5 or 6. That's in its infancy, but that's another area that's really exploding.
Yeah, wow. Such an amazing field.
It is. You can't keep up. Yeah,
No,
it's
no, you
fun.
can't.
And I
All
feel
right.
like a kid in a kid in a candy shop. This is just so much fun, and getting paid to do my hobby. So there you go.
All right. I think we have one last question here. Someone wants to know if fasting is used to weaken cancer at all. Is this used in conjunction with any other therapies?
Yeah, no, it probably is. I don't know much about that. I just know that fasting increases lifespan, and one of the reasons is it decreases oxidative molecules, which can cause DNA damage. That's pretty much the extent of my knowledge there.
Yeah. Okay. That's fine. Um, we have several comments here thanking you very much for the presentation, for making it understandable to people, and just in general for your comments that make it, as I said, make it understandable. So thank you very much for all of that. And then just again, as a reminder, this recording, this presentation has been recorded and it will be available in about a week on the University of Minnesota's OLLI YouTube channel. So that's OLLI. If you go to YouTube and you search for the University of Minnesota OLLI, you can find this presentation as well as previous ones on there for your viewing.
Okay.
All right, so we are
All
out
right.
of time. Thank you so
Thank you.
much, Dr. Pennell, you, the
Yep.
audience, as always, for your
All right.
interest.
Bye-bye.
And also thank you to our sponsor, the University of Minnesota Alumni Association. I do want to say a special thank you to Grayson Simmons for his techno wizardry in producing the series. A big thank you to Steve Benson for finding and booking our amazing speakers. Thank you to Carmi Blyfus for keeping us organized. And a big thank you to Kate Schaefers and Brianna Smith for their outstanding guidance and leadership of the OLLI program. This concludes the At the U series for this year. Please join us next spring for another fascinating look at what faculty and staff at the U are working on. Thank you and bye-bye.